Hello, everyone, and welcome to this webinar, which is focused on stem cells in Parkinson's disease. We will actually focus on stem cell therapy. Stem cells have a role also in research in Parkinson's, but this particular webinar is going to discuss their role, a potential role as a therapy in the future. My name is Patrik Brundin. I'm chairing this session. I'm located in Grand Rapids, Michigan at the Van Andel Research Institute. I'm very happy to have three panelists with me today. Before I introduce them, I shall say that I have a financial disclosure to make. I consult for two companies, Living Cell Technologies and Fujifilm Cellular Dynamics, that both work in the cell transplant space. But now I'm going to introduce our panelists. And let me start with Gaynor Edwards, who is a person affected by Parkinson's disease. And can you just tell us a couple of things about yourself, Gaynor?
Yeah, sure. Hello, Patrick. My name's Gaynor Edwards. I am newsletter editor for Parkinson's Movement, and I also founded the charity Spotlight by OPD, concentrating on young onset Parkinson's. And stem cell is our hope.
Thank you. And Gaynor is located in East Sussex, at least according to her webpage.
That's true.
And then we're going to move across the pond to the United States, where we have someone who has British roots. It's Dr. Claire Henchcliffe, who's at Cornell. Tell us about yourself, please, Claire.
Hi everyone. So my name's Claire Henchcliffe. I'm a Parkinson's neurologist. I have a great interest in developing new therapies, including stem cells, and I'm based in New York City in Manhattan at the Weill Cornell Medicine.
Thank you, Claire. And we come back to the UK, and we will now introduce Roger Barker. Dr. Roger Barker is at Cambridge.
Yes, thank you, Patrick. So I am a consultant neurologist at Adam Brooks Hospital in Cambridge, also the Professor of Clinical Neuroscience at the university. I have worked in Parkinson's disease and cell-based therapies for almost as long as Patrick, but not quite. I do quite a lot of consulting with companies that are interested in this area, which is an area that's caught a lot of attention in the last few years. So I advise Living Cell Technologies in New Zealand, Novo Nordisk in Denmark, Sana Biotechnology in the US, Fujifilm Cellular Dynamics in the US, and Blue Rock Therapeutics in the US.
Thank you. So I must remember to mention the sponsors of this webinar. They are the Cure Parkinson's Trust, Parkinson's Movement, and the Journal of Parkinson's Disease, who all help make this possible. And I'd like also to tell everybody who's listening right now that you're able to ask us questions. You can do that through the Q&A, this little window that pops up. If you click at the bottom of the page on Q&A, there'll be a little window where you can write questions. You can also go to the chat, where it's possible for you to direct questions directly to the panelists or to the whole community that are listening. And the chat gives you the option of all panelists or all panelists and attendees. So those are two good ways of contacting us. I will try to get you involved when your questions come. So bear with me. It may take a few minutes before I can integrate your questions. But I'd like to start off by asking Roger, can you tell us as a group what types of stem cells exist and how are they different from each other? Because in the community, I think people often confuse different types of stem cells.
Yeah, it's a good question, and it is very confusing because essentially a stem cell is defined as a cell that can divide and make another stem cell, as well as turn into some other type of cell. So any cell that has the capacity to proliferate and form more stem cells can, in theory, be used also to treat Parkinson's by being converted into a dopamine cell. Now, the reality is that there are actually probably only two types of stem cell that really have a realistic hope for being used in Parkinson's in the foreseeable future. These are stem cells which come from embryos, so the spare embryos from IVF programs, so-called embryonic stem cells, ES cells. These are the stem cells, as I say, which can be turned into dopamine cells. The very other type of stem cell, which I think has the great potential, are made from adult cells. So this is where you take a cell from a patient or from a person, a skin cell, a blood cell, and you reprogram that cell into something that looks like an embryonic stem cell. And that cell in itself can then be turned into a dopamine cell. And those cells are called induced pluripotent stem cells, or iPSCs. Now, there are lots of other stem cells, as you say, Patrick, but none of those really have the potential to make proper dopamine cells. And I suppose the one which crops up most are things like mesenchymal stem cells that people can get out of adults, often from bone marrow or fat and such like. But the evidence that they can actually make the stem cell turn into the nerve cells that we need to treat Parkinson's isn't really there. So I'd say whilst there's lots of different types of stem cells, and every single person has stem cells, you know, our bone marrow is full of stem cells, our gut is full of stem cells. Really, for our discussion tonight, I think we'll be concentrating on two major types of stem cells, those that come from spare embryos, ES cells, and those which are reprogrammed from adult cells called iPS cells.
So that's helpful. So that means that when people read on the web that there are opportunities to save umbilical cord stem cells or save baby teeth and extract stem cells from these, that there should be a certain level of skepticism about the relevance, at least today, to Parkinson's.
Yeah, I would think so. I think the field has advanced to such a stage, as we'll discuss in the next hour, that you know, embryonic stem cells and iPS cells are about or have already entered the clinic. So I think these other sources of cells may have a future, but at the moment, I would certainly not encourage anybody to invest any money in banking cells or thinking that those cells are going to be superior to anything we've got at the moment.
So Claire, Roger mentioned that one can sometimes get stem cells from the patient, him or herself, and that's what we will call autologous stem cells. How do they compare to those that are obtained from another individual? From a clinical standpoint, what are the opportunities and the challenges with this?
Great question. I think there are probably pros and cons to both. I think that we stand probably to make more progress in the immediate future using stem cells that are not taken from the individual who's going to receive them. But I think the great attraction of getting your own cells has to be that when you're thinking about issues like how are the cells going to engraft? How are they going to survive? Are they going to be rejected? Is there a chance that a transplant could be rejected? Then we can avoid a lot of that discussion and a lot of the concerns around there by giving people their own cells. Now, it's more complicated than that. We all know that if you're transplanting a kidney and that's not immunologically matched, of course, people have to have quite heavy-duty immunosuppressive drugs. We certainly like to avoid that going ahead. It's more complicated in the brain. There's a certain degree of protection in the brain, but it's not absolute. And I think the field has gone through different researchers thinking, well, maybe we don't need to have immunosuppression. And they certainly see some cells surviving there. But I think the current thinking is if the cells are not matched, that there should be some immunosuppression involved. So that's a little bit of more of a burden on the patient. And then I think when you think about what Roger was saying about the possibility of taking someone's own, for example, skin cells and making these induced pluripotent cells that, as Roger said, they look to all intents and purposes like the human embryonic cells. It's very attractive, but those cells go through a certain amount of manipulation. There may be a problem that's intrinsic to those cells about, that has to do with why someone has developed a particular disorder in the first place. And then I think there's also a big practical issue. If you're if for each person who's going to get a stem cell transplant, if you're developing their own lines, that's a lot of work. That's a lot of heavy lifting that has to be done in terms of testing those cells, in terms of quality control, and so on.
Yeah, so we should perhaps mention for the audience that the discovery of the induced pluripotent stem cell is so important. It was done around 2006 that it led to the Nobel Prize for Shinya Yamanaka and for Sir John Gurdon, who had done some of the work preceding that particular discovery. So it's an amazing development, and it's hard to imagine. It is only 13 years ago that this happened, and already, as you will hear later during this hour, these cells are already in clinical trials. So I just want to then bring to the point of discussing, we talk about dopamine neurons all the time, but discussing a little bit the idea that perhaps stem cells could be used to provide trophic factors. These are growth factors that have been shown in various experimental settings to protect nerve cells. What do you think of this concept, Roger? I know many companies are saying, well, we have great stem cells. They don't produce dopamine neurons, but they secrete these growth factors.
Yeah, it's a difficult one to answer, really. I mean, I think you're absolutely right. I mean, stem cells can be thought of as either replacing what you've lost, as we've discussed, or as we will come on to discussion here, replacing the lost dopamine cells, or they can be used to produce things which are beneficial to the brain. So part of that can be growth factors, but also they can produce other factors like factors that suppress the immune system and inflammation, which has also been implicated in Parkinson's disease. So one of the arguments has been that they produce growth factors and these other factors, all of which is a good soup to protect the dopamine cells that are there. The problem, I think, is that that's not been proven really to the same extent as cell replacement therapy with dopamine cells. So if you look at the field of growth factors, fertilizer for dopamine cells, how has it panned out over the last 20 years by just giving growth factors, not actually using cells to deliver them? The story has been a very mixed one. And as many people will probably know, there was recently paper on a growth factor called glial cell line derived neurotrophic factor, GDNF. Wasn't delivered with cells, it was directly infused into the brain by the group in Bristol in the UK, and that didn't show any benefit, and that came on the back of other studies that showed no benefit. Now, that's not to say there isn't a future for it. It may be that, of course, if you're going to use cells which release growth factors or just use growth factors in themselves, you have to be a bit more specific about when you use them. You may have to use them in very early stage disease when patients have fibers and cells that you can rescue more easily than when they've had it for a number of years.
So could I ask you there, because that's an interesting question. So when you say very early, what do we know about when the cells actually disappear in Parkinson's, and a little bit about that process? Does it start at the cell body, or does it start at the terminal end?
Yeah. Well, I think you'll get different views from different people, but my own view, and I would say probably the consensus at the moment, is obviously you have the cell, the dopamine cell body that sits on the stem of the brain, and that sends a long fiber up to the striatum where it releases dopamine. And I think most people would now say that the problem starts at the distal end, where the dopamine is released at the nerve terminal. And then as that dies off, then the cells themselves die off. So rescuing those terminals will be the critical thing. And most people would say by the time you present with motor features of Parkinson's, overt motor features, tremor, slowness, things like that, you've probably lost at least 60, 70% of those fibers. So, of course, by the time people are therefore being seen for the first time by the clinical teams, they've got disease which has actually progressed a fair way down the path with respect to the loss of those nerve cells. So, of course, if you could get earlier and try and pick people up before they had those problems, they may be the ideal group to try and rescue it with these growth factors, with these fertilizers for dopamine cells. But I think, picking the optimal time is going to be, tricky. And I still don't think we know how to do that. There are obviously ethical issues with if you're going to use a cell which produces growth factor, that, you want to use it in newly diagnosed Parkinson's disease. Is that a safe thing to do? Would people, patients be accepting of having an experimental therapy when they've only just been diagnosed? And I think one of the critical factors, which is true for all forms of medicine, is not just whether you can put the things in there, but can you produce the right dose? So I think a lot of the problem with the trials to date has been that there has been underdosing of the patient. And if you imagine that you had a chest infection or a pneumonia and someone gave you an insufficient dose of an antibiotic, your chest infection wouldn't get better. You could then infer that antibiotics don't help chest infections, and you could abandon that whole therapeutic approach. So I think there are unresolved questions around growth factors empirically, with that whole field. Does it have a future in Parkinson's disease? And that therefore puts, I suppose, an even bigger question mark over whether using cells to deliver growth factors is a sensible thing to do at this stage.
I think that's a great summary. So that leaves us with the idea that one should actually convert the stem cells into dopamine nerve cells, and those are the cells that one will transplant, and they will replace the ones that are gone. So can you tell us a little bit about what are the crucial features of the dopamine nerve cells that they have to express for them to be effective, at least in animal models of Parkinson's?
Yeah. So I think there's a sort of assumption that there's only ever one type of dopamine cell in the brain, and that's the dopamine cell that disappears in Parkinson's disease. So actually any dopamine cell will work, and that is clearly not the case. So if you really want to do this job properly and repair the brain around the replacing of the dopamine cells, you've got to put back the dopamine cells which are of the type that are lost in Parkinson's disease. So that is a specific type. They come out of a structure, as I say, in the stem of the brain called the substantia nigra. They're called A9 because there's different A numbers corresponding to different types of dopamine cells. And we know from preclinical work that if you don't make proper A9 dopamine cells of the type lost in Parkinson's disease, they don't work as well. So the first thing you're really going to have to do is you're going to have to make a dopamine cell that is of that type. Now, that is the beginning, because what you also need to do is you need to make cells in enough numbers. So in the human brain, you probably have about 500,000 dopamine cells on either side of your brainstem. And as we've said, you probably lose, let's say, half of them in Parkinson's disease before you present. So you've got to at least make a couple of 100,000 of these cells. You've got to make it.
Will all of them survive? If you make a couple of 100,000, if you implant them, how many will be alive, do you think, in the brain?
Currently, probably only about 5 or 10% of the cells. So you'd probably have to put in a couple of million, which sounds like a lot, but given how many trillions of cells you have in your brain, it's probably not that out of the question. But you are going to have to make enough dopamine cells. So first, you've got to have to make a lot of cells, or enough cells. They're going to have to be the right type of dopamine cells. They're going to have to make connections with the brain. There's no point putting them in the brain if they can't send out little processes and connect with the brain. They're going to have to release dopamine, and they'll have to release dopamine in a way that resembles that which we see in the normal brain. So there are quite a lot of things that have to be done. And one of the problems in the field is that people can sometimes make slightly premature jumps and say, well, I've got something that roughly looks like a dopamine cell, but it's not a proper dopamine nerve cell. It's not of the proper type. It doesn't make connections. And therefore, we know from preclinical work that they don't help animal models. And we know from a long history of not stem cell transplants in Parkinson's disease, but cell-based therapies which have not fulfilled those functions, that they don't work very well. So when I first came to the field in the 1980s, there was a whole trend to use cells taken out of a gland near the kidney called the adrenal. They didn't work preclinically because they didn't survive, they didn't turn into nerve cells, and they didn't make connections. And it was eventually established that they didn't work in patients either. So I think you have to be very critical of the preclinical data and make sure you've got the right dopamine cell to then translate into the clinic. And that, I think, is where we are at the moment. I think there are several groups around the world and several companies that now have the protocols in place for doing that. I wouldn't say they're optimal. I still think we can do better, but they're probably good enough to do the job that we need them to do at the moment.
I think it's fair also to say during the last decade, we as a community, the research community, has defined really what are the molecular features you need. And you can almost go in and test a panel of markers and you'll say, this is the cell that is likely to work, which we certainly didn't know at the beginning of the stem cell era in the early 2000s.
Yeah, and I think also because there's been lots of premature claims, I think, we keep refining what you have to have. So you're absolutely right. I think, 20 years ago, it just had to vaguely look like a nerve cell and have one marker. Now they have to have a panel of markers. They have to have connections. They have to release dopamine. They have to have electrical activity. So I think we're much more stringent about what the cell should have in order to translate into the clinic, which is good, I think, for the patient community and for trials, because we're not trialing things which really haven't got any hope of working.
So I'd just like to tell everyone we've got a couple of questions coming in the chat, and I'm going to get to them soon. But I'd right now like to turn to Gaynor and ask her, imagine now that there is this perfect stem cell-derived dopamine neuron that Roger described. What do you think would be the optimal patient, and at what stage should the patient be treated, in your view?
In my view, in my personal view, now please, Roger. I don't understand why there's a feeling that patients have to get worse before you can actually give them, implant a stem cell. Sometimes, because you don't want to get too bad. You want to be caught as soon as possible so that you can rebuild. Surely, is that not? Does that not make sense?
I think that makes sense. Actually, I had a question from a journalist in Sweden about 12, 15 years ago that was exactly like that. She said, why don't you transplant early in the disease? This was in the days of fetal cell transplants. And obviously the answer was that it was considered ethically questionable to do an invasive procedure in a person who still had lots of benefit from their medication. But one can take your view too, which is perhaps the transplant is more likely to work early in the disease and be better integrated and prevent disability rather than try to reverse disability.
Well, how long did it take for those initial people to sort of, knock at the door and say, this is working for us?
Oh, the time from transplantation to the first beneficial effects was years. Well, there might be subtle effects in the first year, but it took 3-4 years before the full effect of those immature cells being transplanted appeared. So,
I think this is where the field. I think when you're doing a first in human experimental therapy, I can understand the ethical committee's reservations about offering this treatment to people who have a lot to lose and already have a lot of therapies which are there. But I completely subscribe to this view that I think if these early trials are successful, so we don't see complications and we do see benefits, there is absolutely no reason this therapy couldn't be moved further and further back and be used ultimately as, first-line therapy in patients to prevent them developing any of the complications from the medication. So ultimately you could say, well, I've got Parkinson's, I have a stem cell transplant. I don't need to take any drugs. I don't get any complications from the drugs. And therefore you haven't cured people of the condition, but you've changed the complete natural history of treated Parkinson's. I think that will happen, but at the moment, I think it would be a brave ethics committee that would allow you to go into de novo patients with this type of approach.
So one of the questions we got is from Hugh Johnston in Toronto. He writes, what types of Parkinson patients will cell replacement therapy be directed at initially? And perhaps, Claire, you want to say something about that? I know you're working in a clinical program right now where you're recruiting patients. Planning to.
We're almost there. So I think this goes back to what was just brought up actually by Gaynor and by Roger. And I think that in the beginning, we are likely going to be looking at people with more advanced Parkinson's disease, where, as Roger was saying, in early Parkinson's, you really have so many options, and we haven't yet shown that these are procedures that are efficacious and safe, and we still have a lot of work to do. And so I think that initially we're going to be looking at people in the first in human trials, certainly, who have a response still to medication, but where the medication is not really holding them very well. So people who have fluctuations through the day, people where their Cinemet, their carbidopa, levodopa will kick in. So we know that they have a response to dopamine, but maybe it's not lasting long enough, and maybe they're getting other complications from that too. So I think that's where we're going to start. But I have to say, listening to what Roger said, it's nice maybe to draw a comparison with some of the surgical procedures that we already have, where we know that they're efficacious, like deep brain stimulation. And when we started out with that, I remember when I was training, and it was all just starting and very exciting, but we were so restricted in the sorts of people, the sorts of patients who we would think were good candidates for deep brain stimulation. And now if you look at what's happening, we've expanded the age range, and we're thinking about moving that into earlier and earlier Parkinson's disease. And so I think that the beauty of what Gaynor brought up is, why not go early? You could really, if we can show that this works and it's safe, we could help people to avoid all of those exposures to medications with their short-term side effects and their longer-term effects as well.
So that was actually the follow-up question from Hugh Johnson. He asked, what is the timeline like for wider adoption of the treatment, assuming the current trials or the planned trials work? And it's fair to say this is not going to happen in a couple of years. It's going to be a decade before something that is tested beginning today actually will be adopted wider, assuming everything goes really well, right?
Yeah, I think assuming that things go well, it's going to be iterative. We'll make steps. We'll be able to expand the age range, we'll be able to expand perhaps the time over which people have had Parkinson's and the types of symptoms that they have. And then the question is, when are we going to be ready to move into earlier and earlier Parkinson's disease? I think that's going to take a while. I mean, it took, 15 years or so for people to start talking about that in deep brain stimulation. I think we understand a lot more about the processes at play now. So maybe, hopefully not quite that long, but it's going to be iterative, I think.
And you mentioned something. Oh, go ahead, Roger.
I was going to say, I think there are two issues there, really. One I think is that one of the difficulties with cells as opposed to deep brain stimulation comes to a point you made earlier, Patrick, is that the cells don't work straight away. So one of the difficulties is always knowing when they've had their maximum benefit, which takes a while. So you can't assess at 6 months, 1 year, have I done enough? You're probably going to have to wait longer. But that then creates a tension, which is also another reason why I think it might move quicker, is that certainly in the last 3 years or so, this field has attracted a huge amount of interest from biotech, pharma, and so there's a lot of money going into this. And obviously, when you sit on that side of the fence, waiting long periods of time is not something that you do. So I think there's going to be this tension in the field between moving at a speed which is acceptable based on cell-based therapies seeing effects, versus this sort of a sense that we have to move quickly in order to get money back on our investment. So I think it can work both ways. I think things could move quite quickly, and this idea of moving to earlier stage patients may become something that's trialed sooner than we think, because I think the companies can see that there's obviously great mileage in and a logic in that. So I suspect that once the early trials, assuming they show safety and that nothing adversely happens with these cells and there's some signal of efficacy, there may be a big push not to develop bigger trials in more advanced cases, patients, but actually to go for an earlier use of the cells in terms of the disease course.
So we have a question about how, it says, can the stem cell treatments roll back the Parkinson's clock? And what types of symptoms will be improved and which ones are unlikely to be improved? Do you want to address this, Roger and Claire? And maybe Gaynor, you want to chip in and tell us a little bit what the expectations are in the community? Do people think that stem cells are the panacea that will affect everything and bring you back to a pre-symptomatic stage?
For hope.
Is it a realistic hope?
I hope so.
What do you say, Claire?
So I think to begin with, what Roger was saying is absolutely correct. We're going to be looking at rebuilding dopaminergic networks. So I think we have a fair idea of what we can be looking for there. We can look for help with the slowing, we can look for help with the fluctuations, the ons and offs that people get. We can look for help with the rigidity, maybe the tremor and some of the other motor symptoms. And we can look for help as if the cells work well, people might even be able to, they could reduce their medicine. They might even be able to get off their medicine. So you could think about reducing all of those off-target effects of medications and lighten the load very much. But then the question is, we all know that Parkinson's is so much more than a movement problem. And so the question is, what's going to help some of the other symptoms? When you think about, again, I'm going to come back to the deep brain stimulation. And there's definitely evidence that although we thought at the beginning we were going to just be looking at movement symptoms, it does seem to help a lot more than that. It can help people with sleep, and it certainly helps people's quality of life. So I think as we're testing these cells, we need to cast a pretty broad net and see what they can do. But I think the real fascinating stuff for down the line is what can we do outside of movement? And is this going to take different types of cells? Is it going to take combinations of cells or combinatorial approaches? Where do we have to get the cells in? How do we address symptoms that are more widespread in terms of the underlying mechanisms?
So I suppose...
I was just going to ask Gaynor, how do you feel about that answer? Is that a disappointment to hear that it's not obvious that it's going to help several symptoms, or
I would wonder again whether what happened with those original patients, did that have an impact on their non-motor symptoms?
Maybe you want to answer that, Roger.
Yeah, I mean, I think, so I think the first thing is I don't think it'll roll back Parkinson's disease because the disease process, whatever drives it and whatever causes it, it carries on. So the accumulation of this protein alpha-synuclein is something which these cells will not be stopping. So I think it's always very important to understand these are not a cure. I think the way I always think about this is you know, what happens when patients take dopamine drugs? So, you know, in early stage disease, you take your L-dopa, Sinemet, Madapar, whatever. What sort of state do you get to? Well, you get pretty good. And that's not just dealing with motor side, that's non-motor side. So my own view is that these cells are targeting dopa-responsive elements. Now, that will include some cognitive aspects. So some thinking problems with Parkinson's disease relate to actually dopamine and abnormalities in dopamine networks. So I think it'll improve people's thinking. As Claire says, I think if you get better motor control, you get better sleep. If you sleep better, your whole day's better. So I think that will improve whole things around quality of life. So I think there are direct non-motor benefits which you'll get from these therapies. Similarly, by not taking all the medication, you avoid all the non-motor complications that you get from taking the medication. So some of the psychiatric problems you can get from the medication. So I think whilst it's clearly targeting only one part of a more distributed pathology, it could have implications and ramifications across lots of different symptoms and signs. And you're probably a better expert than me on this, Patrick, but certainly in the Swedish patients, you know, it was clear that there are reports that the non-motor aspects of the disease improved. I think the difficulty with those studies were that in the 80s and 90s, when they took place, the non-motor aspects of Parkinson's disease were not something that was very much talked about. People talked about dementia. People talked about some aspects around thinking, but it wasn't a major aspect. It wasn't really recognised as a major
and
aspect.
It wasn't really followed at all in those studies. So there were some very small side studies done on those patients, I think it's fair to say.
Yeah. So I suppose I take a more optimistic view that whilst these cells are not curing and they're not treating the whole problem, I suspect if they work well, they will have the benefits which you see with dopamine medication. And in early stage disease, when people take the dopamine medication, and Gaynor may want to speak to this, I mean, it puts people pretty back to normal. And I would expect that's what the cells will do. So it will be motor and non-motor aspects, but it won't treat everything. And I suppose, you know, those non-motor aspects, for which we do have some drugs, there's no reason, of course, with these cells that you can't combine things. And we may come on to discuss this. So I think it's always important to view therapeutics and experimental therapeutics not in isolation. So whilst people talk about disease-modifying therapies, and they talk about cells as though they're independent therapies you'll have, there's no reason you couldn't combine these and get the benefits from both.
I think that's a great point, Roger, as well, that if people are not having to worry so much about controlling their motor symptoms and worrying about the fluctuations, the ups and downs, the dyskinesia, then it really lightens the load on them. And it kind of frees you up. You know, in my clinic visits, if we're spending all of our time talking about motor symptoms, we can deal with the other symptoms. We just don't have a lot of time or energy left over sometimes to be able to focus on that.
Gaynor, you wanted to say something also?
It was just really that if the stem cell was able to kick in before the negative side effects of levodopa, so, you know, in a young onset, if it could beat the dyskinesia. You know,
that is a great question, actually. So I'd actually like to bring this up a little bit more. So young onset Parkinson's disease, as you know very well, Gaynor, in many cases there is a genetic form that doesn't have the alpha-synuclein aggregates or the Lewy bodies. And theoretically, that group of patient might be the ideal group of patient for stem cell transplants, because there won't be any synuclein aggregates forming inside the transplanted cells, and typically the clinical course in those patients is very protracted and it's very motor driven. And as you probably know, and many of those listening know, there have been articles written discussing whether trials should be focused on people with so-called Parkin mutations. And I think the answer to that is it would be fantastic if that were possible. It's it's practically rather difficult to arrange because there aren't that many patients like this that would be available for those trials. I don't know if you want to comment on this, Gaynor.
Well, is that more because we don't know what all of the gene types, you know, they're not automatically tested, some of these younger people. So, I mean, if they were tested, maybe they would be more available.
I mean, I have to say that in our TransEuro study, which is a study which is not stem cells, it was a trial which we developed about 10 years ago to revisit using fetal dopamine cells. There was a huge discussion actually about whether we should concentrate on Parkinson positive patients to treat. And we weren't going to treat the whole group wasn't just going to be Parkinson positive, but we wondered about having a subgroup of patients. And the problem was exactly as you said, Patrik. There were three problems we saw with it as an approach. One is the number of patients is actually quite small who have problems in both Parkinson genes. You can have a problem in one Parkinson gene, so-called heterozygote. Whether that's important and gives you a different type of disease is debatable. But true Parkinson positive patients are actually quite rare. Secondly, there was an anxiety that if you treat Parkinson positive Parkinson's, and this comes back to a point earlier about commercial development of cells, people will say, yeah, but you haven't treated idiopathic Parkinson's, which is what most people have. So it's a rather niche area you're treating. So it has got limited commercial potential. And the main reason we actually didn't was that there was a concern in some of the earlier trials that the transplants could lead patients to have involuntary movements, to have dyskinesias, so-called graft-induced dyskinesias. And they were very much had only been seen in people who had L-DOPA-induced dyskinesias, so involuntary movements driven by the drugs. And one of the problems with the Parkinson-positive patients is that they have a very marked tendency to develop quite early and quite major L-DOPA-induced dyskinesias. So whilst we built it into our original trial 10 years ago, we practically couldn't find enough patients, and then we argued ourselves out of it. So I think in theory it has potential, but I think it would be quite a hard trial to actually do myself.
So I'd like to bring in a couple of questions that touch upon the topic we've just discussed now. There's one from Deepak Mohan asking about graft-induced dyskinesias. Are they caused by serotonin, nerve cells in the transplant, or other impurities? And there was another question. Let me just scroll up. So, I'll answer one of the questions. I'm getting lots of questions suddenly. How much collaboration takes place in the research? There are a number of different research centers doing stem cell research. Do they collaborate? And I'll answer that relatively briefly, that there is an amazing amount of collaboration in this particular field. And there is a group called G-Force PD, which is focused on global collaboration on stem cell transplants. And there are participants from Japan, from Scandinavia, from the UK, from the United States. And
China last time.
China the last time, that's true. And having worked in different parts of Parkinson's research in my career, I think the collaboration in the stem cell transplant field is really remarkable because people share some of their innermost scientific secrets before they are quite ready with them. And, you know, we have to have good evidence before we put forward our ideas. We have to write about them in scientific papers and publications. But in G-Force PD, people share preliminary ideas and evidence in a very open way. What complicates matters, of course, is that to be able to do these large trials, one needs the financial backing of drug companies or biotech companies. And once you bring in biotech companies, they have to protect their inventions. They have investors who want to have return on investment, and they are therefore not able to share everything. But I still think at this stage in time, the collaboration also between those biotech and drug companies that are involved has been remarkable, and that's a big positive. But let me go back to the question about dyskinesias, and I'd like to open this to anyone who wants to speak on the panel. We heard from Roger that there were these graft-induced involuntary movements in the fetal cell trials, and they basically meant that the fetal cell trials grinded to a halt around 2001 because we didn't understand the mechanisms underlying these involuntary movements. And in some patients, they were severe. In many patients, not, but in some, they were severe. And what do we know today about these graft-induced dyskinesias? And as a second related question, are there other potential dangers with transplanting stem cell-derived nerve cells? And what can we do to minimize these dangers? After all, stem cells divide indefinitely. Is that a danger?
Yeah, so I suppose if I just come back to dyskinesia, so I mean, I would say it's still not resolved entirely as to what causes graft-induced dyskinesias, because one of the big problems in trying to understand them is that they are impossible to model in the lab. So Patrik's developed a model which is sort of similar but isn't the same. So we can't model it in the lab. So we have to go back to what we've seen in the clinic. And I think there are two views as to what's causing graft-induced dyskinesias. One is that it relates to these so-called dopamine hotspots, that you haven't delivered the dopamine cells in an even fashion across the area you've transplanted them. So some areas are releasing dopamine, others are not. I think most people don't really subscribe to that view. And the commonest view is that the dopamine cells which we collected from the aborted fetal brain, those dopamine cells develop right next door to the 5-HT serotonin cells, which have the capacity to make dopamine, but they have no capacity to get rid of it because they can't take it up. So the dyskinesias were driven by this ratio of the serotonin, contaminating serotonin 5-HT neurons and the dopamine cells. Now, if that is correct, then obviously as we move forward with stem cells, the idea of graft-induced dyskinesias will not be one we need to worry about, because obviously the beauty of stem cells is you direct them to dopamine cells, and you only direct them to dopamine cells. And if they don't form dopamine cells, they form something en route to that, but they certainly don't make 5-HT serotonin neurons. So the hope would be that graft-induced dyskinesias, if that theory is correct, will not be a worry with stem cells. The main anxiety from, I would say, from the regulatory point of view, understandably with stem cell transplants, is can you be absolutely certain that there isn't a stem cell lurking somewhere in your transplant that has the capacity to proliferate? And obviously, once it starts proliferating, it will continue proliferating, as Patrik says, and then you have, in essence, a tumor or a space-occupying lesion, a mass lesion in your brain, and that is a problem because you can't get it out. I think actually what's more of a worry for me is I don't think that is going to be an issue because the protocols are so good. And for example, in the Swedish group led by Malin Palmer, they have never seen in their transplanted rats, and they've now transplanted 1,300 rats. So the chances of a tumor, I think, is not going to be anything we actually need to worry about, but we obviously need to pay attention to it. I think the greater worry for me in terms of safety is whether we're actually going to be producing enough of the cells of the right type. So can we be certain, outside of the risks of neurosurgery, that the transplants themselves are actually going to differentiate and turn into the cells that we want them to, with the maturity and the capacity to release dopamine and reconstruct circuits in the way that we do? Because if they don't, then I think that there's two things that can happen. One is you could get behaviors that we don't even know what they are at the moment, because we can't model Parkinson's in the lab. We can only model loss of dopamine. Or secondly, you have an ineffective therapy and it gets abandoned at a level of development which is a bit premature. So I don't worry so much about graft-induced dyskinesias with stem cell dopamine cells. Regulators worry about tumours, understandably so. I think the worry scientifically is whether we've actually got a protocol which will allow us to make enough of the cells in the right way to actually work in the Parkinsonian brain.
Related to that, Gaynor, there is a lot of discussion online in the PWP community about stem cell tourism, essentially people traveling to countries where the regulatory agencies aren't very active or efficient and getting transplants. And how aware are people with Parkinson's that there's a true risk with going to a place that isn't scientifically established? There's a risk that those cells will actually form tumors.
Sorry, who was that to?
That's to you, Gaynor.
Oh,
I don't know, in the PWP community, are people aware of the risks of stem cell tourism? You go off to some remote island where there's not much of a regulatory body, and those stem cells will, just as Roger described, a few of them will still be dividing, and there'll be a tumor forming.
I think it depends how desperate they are and how well educated they are within the Parkinson's community. Those that have actually got to acceptance and can actually confront their condition and think it through properly, they're fine. They're the people that we can work with happily. The ones that you know, ain't just a river in Egypt, stuck in denial. You know, there's more of an issue with them. They're more likely to be desperate and just go anywhere, find anything. They don't want to know too much detail about it. They just want to be fixed.
But I also see in the US, I follow a lot of Facebook groups, several people who show great interest in stem cells tourism, at least on the Facebook stage. I don't know how many of them actually travel to these clinics.
I don't know. It's certainly not something that I'd advocate. And if I heard anyone considering such a thing, I'd be talking them out of it.
That sounds good. So another thing is, if
speak to that actually, Patrick, because stem cell tourism is a real worry, not only for the individuals that get, if you like, sucked into the hyperbole and the sort of testaments which often appear on these websites, and therefore go and have these therapies. But it has the potential to completely derail the whole field. So if someone has one of these therapies, even in a non-licensed clinic, and develop a tumour and die, sadly with this, it will have implications for everybody. So it's not something that I think we need to just sort of acknowledge and leave. I think we do need to be quite active in saying what the dangers are. And the International Society for Stem Cell Research, the ISSCR, for anyone who's interested in sort of trying to understand what a clinic is offering, they have fantastic resources by which to sort of interrogate and assess websites and companies offering such therapies, to try and help support people through that process of whether this is a legitimate centre that you should be thinking about going to have such a treatment. So
the vast majority, if not all of those companies, I would say, offer treatments that are not based on proper science. And they often start with, for example, fat cells that they claim have an ability, stem cells derived from fat cells, to make dopamine neurons, but there's no scientific proof of it. So there are two problems. It's very unlikely to be effective. And the second problem, of course, if there are dividing cells in this cocktail that's injected, it may actually produce a slowly growing brain tumor.
And that has happened in other conditions.
Yes, it has. Yeah.
Injury, you got a spinal cord tumor. There was a child that died in Russia from having stem cells for another neurological condition. And there are people who've had eye-related degeneration, who've had fat cells, who've gone from poor vision to no vision. So as you say, these are not benign therapies. They have the potential to make you worse.
So Claire, I want to bring you in because you are, as we said before, involved in the late planning stages of a clinical trial using stem cells with a very strong scientific basis, I should add. But somebody asked, what are the challenges of using induced pluripotent stem cells or directly programmed dopamine neurons as transplants in Parkinson's patients? And I can maybe explain briefly a directly programmed dopamine neuron. It's the notion that one takes let's say a skin cell and uses a set of genetic factors and manipulates this cell in the culture dish. So it goes from skin cell directly to dopamine neuron without really being a stem cell. And I can just say the challenge with that is, with the current protocols, it's be very hard to get a high number of dopamine neurons. And secondly, the work that's been done so far has not illustrated or shown that these dopamine neurons that are obtained are really true midbrain dopamine neurons. There's very little literature, so there's a lot of work to be done there. But with induced pluripotent stem cells or human embryonic stem cells, how can we make these competitive and manufacture these in large quantities so they can be distributed to many centers and placed in many patients? And another question here is, should the approach be, in the case of iPS cells, to use a starting material, a cell from the patient, him or herself? Lots of questions there.
Loads of great questions there. In terms of, you brought up something interesting, which has to do with very practical applications and development of treatments. And it applies to drugs and any sort of treatment as well as stem cells. And that has to do with being able to manufacture the cells in enough numbers. Many of the cells, the cell lines that are used obviously can be manufactured, can be grown in almost limitless quantities. And I think if groups are working on cell sources where they're able then to cryopreserve the cells, you can kind of see how you can end up with a bank which is almost off the shelf. It could be shipped cryopreserved. It just takes the, it takes a lot of the pressure off surgical programming and surgical scheduling. And it means that essentially you can do all of the quality control and the testing for efficacy and the testing for safety in the same sorts of cells that are going to be used for transplant in the actual patient. So I think that's huge. And to me, that's what gives the human embryonic stem cell lines a real sort of head start at the present point. Taking people's own cells really means that you're looking at, like I said before, you know, developing a different line or a different set of cell lines for each patient. So it just makes it logistically incredibly difficult to do the amount of testing, get the amount of quality control done, get the amount of efficacy testing, and so on done in all of those stem cells.
Could I just interrupt Deepak Mo and ask, how expensive are these autologous iPS cells? Do you have a sense of what that would cost per patient?
I don't actually. Does anyone else on this webinar know how much?
About £1 to £2 million, dollars or euros.
Then I would take dollars because that's cheaper.
Yeah.
It's seven figures, whatever you want to put it in. And to be honest, this was so with the Japanese program, where obviously iPS cells were invented or discovered. They were using this approach to treat eye disease. So they were using the patient's own cells for a program to repair patients' own eyes. And after they did one patient, they realized the cost of this going forward was just be prohibitively expensive, because each person would have to have the cell manufacturers, you say, Claire, then they have to have all the safety testing and all the quality control. And that would have to be done with every single patient. So it's incredibly expensive per patient. So currently, financially, unless you could license the process as opposed to the product, it's not going to be a viable option economically going forward.
But Claire, do you really think that having autologous cells from the patient, him or herself, is necessary? Or does the biology suggest that we can transplant cells from people who are genetically different to each other, and those transplanted cells will still survive in the brain?
Well, we know that we can do that. We know that we can take non-matched donor cells, as has been done back in the 80s, 90s, and those cells will survive. And if we provide immunosuppression, maybe they survive a little better as well. So I don't think it's necessary for people to have their own cells, but it's very attractive in many ways. And so I think, although we're not there yet in terms of logistics, but I think in the future, these are going to be interesting options, including what you brought up, the inducible neuronal cell, where you do a direct reprogramming and go from, say, skin to neurons. So I think those are really exciting possibilities, but I don't think we're there yet.
Well, of course, people also think about not just making skin cells directly into dopamine cells, but the ultimate would be, why not just take spare cells you have in your brain, namely the glial cells, and directly convert them into dopamine cells? So never transplant anything, just simply re-educate the brain and reprogram the brain to produce the cells that you need. So
whilst this sounds like science fiction, there are actually scientific laboratory programs testing this in animals at this time. Of course, it's a very early stage, but there the idea is just using a needle to inject the genetic factors and using viruses to put those genetic factors into the glial cells and then make them into the missing dopamine neurons. But I think there are many experiments that need to be done before that ever could be considered clinical. So one question that's popped up is, could the stem cell therapies be combined with other new innovative therapies? For example, there's this whole program of immunotherapy where the target is alpha-synuclein, the protein that forms the Lewy bodies. Would it be valuable? Is it possible in the future that one will treat people with an alpha-synuclein immunotherapy and with a stem cell transplant? And what could that achieve that either one alone would not achieve? Claire?
Yes, so I think That's incredibly exciting, actually, and the idea of starting to combine different approaches is really, really compelling. What could you achieve with cells in combination with an anti-alpha-synuclein therapy? Well, you could take someone with diagnosed Parkinson's, you could restore their motor function, so they really don't have to worry about where their dopamine is coming from. But at the same time, some of the things we were talking about earlier, like the spread of Parkinson's to other areas of the brain that might cause problems with cognition, problems with mood, etc. Using an anti-alpha-synuclein therapy, you may be able to just damp that down or maybe even stop it. So you could be able to overcome some of these more widespread effects of Parkinson's, and at the same time, in the motor system where you've already got damage, be able to restore that.
So, Gloria, Gaynor. Gloria, that's Gloria Gaynor. That is a Freudian slip of the tongue. Reach out and I'll be there. So, Gaynor, could you just say something about what you, as a representative for the PWP, also think about stem cell therapy in terms of savings, I believe, at the societal level, that we shouldn't forget that there's not just reducing human suffering, but it's also a question of smart science that will save a lot of work-life years, reduce burden of care, etc.
Absolutely, because, you know, we're talking about the cost of it, and you know, granted, I realise it's not a cheap treatment, but you know, going forward, what is the saving? Because if we're looking at young onset people that have 30 years, 40 years, 50 years to live with this condition, depending on when you give them the treatment, that's an awful lot of savings in potential or benefits. If you know, if they're based in the UK, for example, NHS hospital visits, they stay within work. You know, the quality of life and everything there's a price to it all, isn't there?
And I think you're absolutely right about this. And we often think in the very short term, and you know, I don't want to sound cynical, but some drug companies also think in terms of the value of having patients on medications for a very, very long time. Whereas with stem cells, there'll be a one-time treatment perhaps, and they may never need to have that treatment again. And I'm not saying that companies are ignoring stem cell therapy because of that, but they obviously look at that calculation differently to one where they know they can give a drug that's worth, let's take immunotherapy. I'm presuming immunotherapies, if they ever were to work for Parkinson's, they would require administering an antibody for hundreds of thousands of dollars per year for 20, 30 years, which is an attractive financial proposition for a drug company.
It is, but I think one of the reasons that drug companies have, or companies got interested in cell-based therapies is, it comes back to a point you raised earlier, that the actual production of these cells, so to take a stem cell and turn it into a dopamine cell, takes about 2.5 weeks. And it's not a particularly labor-intensive process. So the manufacturing of large numbers of cells can be done relatively cheaply, which therefore means, of course, if you offer a reasonable markup on that price, from the manufacturing, you actually have a huge margin of profit on something that you can make quite easily. So, you know, most people won't understand that it takes 16 days to make a dopamine cell. That sounds like quite a long time, but people who are thinking of making other cells for the brain, sort of these supporting cells, glial cells, those protocols are 180 days long. So trying to manufacture something for 180 days and control that process and reproduce it, that's very difficult. 16 days. You can start from a 6-well plate, so a small little plate, but it's that big, and within 16 days you'll have made enough dopamine cells to treat 500 patients. So that's just me sitting in a lab doing it myself. So
I doubt it would happen if you did it, Roger. But
yeah, well, I'm trying to play things that perhaps people who know me wouldn't trust me, but the principle is there, and so I think manufacturing costs are much less than people I think originally thought, which is why it's attracted a lot of interest from companies.
So we are actually drawing to a close, and I really want to thank Gaynor, Claire, and Roger for great answers and a great discussion. Also thank all of you who contributed with questions. We weren't able to answer all of them. It's been a remarkable number. I know we had about 110 people or so registered in this webinar, And it's going to be available on YouTube, I believe, tomorrow, so you can see it again. I know some of you had problems hearing us, and if you did, hopefully the YouTube recording will be a little bit better. But we're very grateful to the panelists and to all the listeners and people posing questions. And we will be returning in about 3 months from now with a new webinar, with a new topic, and we welcome suggestions for anything you want to discuss with us. So thank you, everyone, and bye-bye.
Thanks very much. Bye.
Bye.
Are we off air?